Hi everyone! Welcome to Activity 9. Today we are going to perform Hematocrit, or otherwise known as packed cell volume. These are the materials that you will need for the procedure. First, we have our sharps container as a receptacle for our used lancets. We also have capillary tubes for our blood sample. We'll also need cotton and 70% alcohol for disinfecting the area of collection. And we have clay sealant, paraffin wax to prevent our samples from spilling. We also have our lancets to puncture the site of collection. We will also make use of our readers such as the circular or rectangular reader to determine our hematocrit results. Finally, we will also be using a microhematocrit centrifuge to spin our capillary tubes for 5 minutes. These are the following steps in order to perform the hematocrit method. First, you have to perform capillary puncture on the third or fourth finger of your patient's non-dominant hand, observing proper disinfection of the site. Using the lancet, perform a quick firm stab, wipe the first drop of blood, and then collect your sample. You have to fill 2/3 to 3/4 of your capillary tube. Invert to mix. After which, you have to get a dry cotton and then cover the site of collection, instructing your patient to press until bleeding stops, and then seal your capillary tubes with clay sealant and paraffin wax. After sealing the capillary tubes properly, they are now ready for spinning using the microhematocrit centrifuge. Open the microhematocrit centrifuge and lift the lid, after which remove the cover of the gasket. Position the tubes with the clay end against the gasket. Make sure to balance the tubes by placing 2 tubes opposite to each other. Also, take note of the number where you position your tubes. This is to avoid interchanging of blood samples if you have 2 or more patients sharing the same centrifuge. After making sure that all of the tubes are already balanced, place the head cover of the gasket and make sure to secure it tightly and close the top. Spin the microhematocrit tubes for at least 5 minutes and make sure that the centrifuge is in complete stop before opening it. Now let's go and try to read our hematocrit results using the rectangular reader. Position the capillary tube on the groove found on the plastic plate. Align the interface of the clay end over the bottom black line found on the plate. Make sure that the black line is above the clay. Move the capillary tube to the right until the top portion of the plasma is in line with the diagonal black line found above the plate. Place the plastic indicator line to the center of the tube until the line intersects between the buffy coat and the packed cell layer. Get the reading at the point where the indicator line is intersecting with the calibration found on the right portion of the plate. Report the reading in percent.
In this case, our reading is 45%. This time, let's try to read our hematocrit results using the circular reader. And let's try to compare if our previous reading using the rectangular reader is the same with this one. Position the capillary tube on the groove found on the plastic plate. Align the interface of the clayed end over the bottom black line found on the plate and make sure that the black line is above the clay. Rotate the bottom plate so that 100% line is directly below the red line of the plastic indicator. Rotate the top plate so that the curved line intersects with the top portion of the plasma found in your capillary tube. After which, rotate both the disc until the curve intersects between the buffy coat and the packed cell layer. The result is read from the point of the scale directly beneath the red line of the plate's indicator. Report the reading in percent. So that's it. That's how you perform reading of your hematocrit results. Thank you so much for watching, and I'll see you next time.